Thank you, Brad, and thank you. Thanks for having me, and thanks for inviting me to this exciting workshop. Unfortunately, I won't be able to. I need to ask the person in the back to change my slides. But yeah, I'll be talking about pharmaceuticals in the aquatic environment today. Moving a little bit away from. I'm a regulator, but I'll be presenting some case studies that we've done here at the EPA. So next slide, please. So, but I thought for some of you in the audience, you might not know that EPA has a Emerging Contaminants Program. So it was launched around the time that I started 2 years ago. And the aim of the program is to monitor and assess environmental quality of water, land and air, and identify and assess the environmental and human health risks associated with these chemicals. But overall, the project is there to, the program is there to reduce the risk and prevent harm to human health and environment from pollution and waste, but also to increase EPA's understanding of these emerging contaminants and develop better environmental standards and guidance for management. Next slide, please. But yeah, today's topic is pharmaceuticals, and it's definitely an emerging contaminant. And I hope that you agree that it's a growing global problem. And next slide, please. I'll put a number to a problem. This is a diagram from Bernard et al. From Ecology Environment 2017, where he mapped proportional change since 1970. As you can see, the global pharmaceutical consumption is overseeding even human population impact from increased human population and atmospheric CO2, and it's increasing at the rate more than double than any other global change factor. And next slide, please. But it's important to be aware of where are these pharmaceuticals coming from. So when we're looking at them, how could we solve this problem? And they're actually coming from a few different sources. So there's almost a branch that is vegetarian pharmaceuticals and then human pharmaceuticals. And the vegetarian pharmaceuticals could be categorized under aquaculture, companion animals, livestock. And from those, often the pharmaceuticals might be going quite directly to freshwater and terrestrial ecosystems. But then when we look at them, the more traditional, what we can easily think about is the human pharmaceutical pathway. So that is then through households, hospitals and healthcare facilities. And those then go through the wastewater treatment systems and through solid waste disposal. But it's important to be mindful that also septic systems and biosolids could be partially responsible for the release of pharmaceuticals. But overall, we're looking at more than 4,000 active ingredients. So it's a quite of a chemical challenge. Next slide, please. But let's delve into the first case study that I was lucky to be part of. So this was a work that we did March-June 2020, so a year ago. And the aim of the study was to establish background concentrations of pharmaceuticals and personal care products and endocrine disrupting chemicals, EDCs, in freshwater and in biota across Victoria. But also we were interested in investigating differences between upstream and downstream sites and association to the discharge points. Next slide, please. And next, yep, thank you. And here is the map on the right, which shows the different sampling locations. So we had overall 18 sampling sites, which include 3 reference sites, and we were collecting both fish and water from each site. And the design was that we had 3 sites per location. So we had the hotspot was the discharge point. We were not measuring the effluent, we were actually measuring then the river or creek. And that was around the 50 to 100 meters, depending on the access to that spot. And then we had an upstream site, which was approximately 2 kilometers upstream, and then downstream another 2 kilometers. And yet we collected approximately 18 to 20 fish per site. But the guys actually were so successful that I would say I almost had to say like we had to stop because we had a limited budget, but the budget was generous. We were able to analyze more than 300 fish, and we were able to analyze them individually. So we did not do any composite sampling. And for water sampling, we used crab sampling and passive sampling 28 data. So those zero is the day when we deployed the passive samplers, and day 28 is when we retrieved the passive sampler. And then we analyzed the water samples and fish for pharmaceuticals, personal care products, EDCs, and also PFAS, but I won't be talking about the PFAS work today. Next slide, please. So let's move on. So what did we find? So here on the y-axis, you see total pharmaceuticals in micrograms per liter, and the scale is from 0 to 10. And then on the x-axis, you see different sampling locations. So first on the left, you see reference sites, and then there's a Barnaby Creek, Campaspe River, Jackson's Creek, Mullan Creek, and Mary Creek. And then there is the upstream discharge and downstream panels. If you look at then the different different histograms. And then I thought I'll start by listing some of the top 10 chemicals that we were able to analyze. So venlafaxine is an antidepressant, paracetamol is a painkiller, and oxazepam is a medication used to treat behavioral syndromes. And then there is a cephalexin, is the antihistamine. Carbamazepine is used to treat again a medical sorry, now I need to check and look at my notes for that one. Can't find them. Anyway, that one. Sorry about that. Carbamazepine is a treat epilepsy and neurotic pain. Yes, actually, yeah. Carbamazepine will be coming up later as well. And then acetylcholine is an artificial sweetener. So a selection of painkillers and antidepressants, and then also some repression drugs as well. So those 10 were coming as top 10 in terms of concentrations and their frequency. And if you then look at the different sites across those locations, it's clear that there are differences. And the differences are coming from so different SDPs have different levels of treatment. So for example, the Barnaby Creek has a level B treatment, while the Mary Creek has a level A treatment. So the treatment is a clear difference between those two locations. But then another thing that is good to be aware of, that there's a different flow, that different volume of water that they treat daily. And in a Barnaby Creek is around 8,000, while in Mary Creek is more than 2,500. And also the Barnaby Creek, my understanding is that it's receiving combination of domestic and industrial waste. So therefore, that could be one reason for the high load in terms of the total pharmaceutical in the discharge point. But one thing that I think is important to take home from this is that when you compare sites, yes, the discharge point is clearly, we were expecting it to be the hotspot, but if you look at the Mary Creek location, there seems to be very similar concentration upstream and downstream. So it's likely that there are other hotspots or sources around, and it's more diffuse than we have thought about it in the past. Next slide, please. And then we looked at, as I mentioned, we used both crab sampling and passive samplers. So here on the y-axis, we have mean concentrations in nanograms per liter. So it's from 0 to 1,200. And just there's a lot of, as you can see, all the list of chemicals that we analyzed, that we measured 72 in total. And in POCUS, we were able to detect 59, while in a crab sample, 39. So again, take home this is that it's important to use different types of methods to sample, especially pharmaceuticals, because depending on the method you use, here the result would have been quite different looking at all the spot water samples. But then also when we use the spot water samples, they were actually highlighting slightly different chemicals. So a combination of methods is the best approach here. Next slide, please. And here is then a quick table of the pharmaceuticals we measured in biota. As I said, we did analyze more than 300 individuals, but this is now a list of all the fish that we were able to analyze using the edible portion. So here you have on top, there's a blackfish, carp, eel, redfin, then there's a roach and hens. And out of those three, next click please. So we analyzed 22 analytes, but only caffeine, carbamazepine, and venlafaxine were the ones that were coming up that we were able to measure above the limit of reporting. And next slide, please. Let's look at the venlafaxine in more detail. So venlafaxine, here I have summarized results, so measurements of venlafaxine in fish muscle tissue across all sites. And they are first panel is reference, then it's upstream, discharge point, and downstream. And on the y-axis, we have a venlafaxine in wet weight, micrograms per kilogram, from 0 to 150. And if you do the next click, you see that there is, at the discharge point, there was one individual that had 150 micrograms per kilogram in his edible portion, which was quite a high reading. So we had to go back to the lab and double check that it was a real result, but it is. And when we looked at that particular waterway where we detected that redfin actually downstream the fish have venlafaxine as well. So it seems to be that then in that waterway, venlafaxine concentrations are higher compared to the other locations based on this study. And next slide. And we did risk assessment, but I'm not going to go due to the time constraints in details of that. Based on the risk assessment, the risks for human consumption are relatively low, but I would like to draw your attention to that there is abundance of research showing that there's a lot of adverse effects of venlafaxine in non-target organisms, and they vary from high bioaccumulation to reduced survival, to reduce embryo production, and most importantly, also their chasing behavior, and at the concentrations that we have detected in the environment. So definitely there is ways to do more research and have a better understanding of the risks to the human health, but also to the ecosystem. Next slide, please. And now quickly, I'm just going to show you a few I would say, this is a new study, case study on emerging contaminants in recycled water. We just got the data, and I'm very excited about, so I want to share some of the preliminary results from this work. It's funded by DELP, Department of Environment, Land, Water and Planning. We sampled 30 SDPs across Victoria, and this study was done this year. So during one of the lockdowns, I'm losing count of how many, what was the number from April to June, so maybe the fourth lockdown. And the site selection for those 30 sites was based on the 2019 desktop assessment done by Nick O'Connor and Daryl Stevens. And those sites were selected on, so they went through all the SDPs on the water corporations in Victoria and looking at the different flows those sites have, what kind of catchment types they have, and then the treatment processes, and then based on that information, created a different catchment risk scores from low, medium, and high, and then they have 11 treatment trains. So based on that information, next slide, please. Then we selected 30 SDPs, and we aim to have replication per treatment train to get a better handle on how these SDPs are actually treating pharmaceuticals in particular. So we're measuring influent and effluent water at these locations. We were able to do composite sampling, but again, we used both crab and passive samplers. And then that work is still underway, but there is a photobacteria test and antioxidant assays that are done in University of Melbourne. Next slide, please. And here is one of the first kind of overall pictures of what we are seeing in our effluent samples. So there is an influent and effluent. On the top panel is the influent, and I want to next click, please. I just want to highlight that the scale between the top and the bottom is very different. So we're looking at mean pharmaceutical concentrations in micrograms per liter, but the top panel is from 0 to 300, while the bottom panel is from 0 to 2. So overall, what we are seeing is that the pharmaceuticals, if you look at the top maximum concentrations, they are treatment is treating the wastewater relatively well. But what we know about pharmaceuticals is actually often the low concentrations that really matter, and it looks like the effluents are still they contain a variety of pharmaceuticals. Next slide, please. But I wanted to highlight some of the, I would say, examples here where we're seeing a good removal, and then some chemicals where we're not seeing such a good removal. So on top there's acesulfame K, there's caffeine and paracetamol. And if you look at this, now influent is purple and effluent is blue. For example, for paracetamol, we're going from 400 to less than 5. So that is a very significant reduce in concentrations that are coming in and then coming out. But then the other that are not that greatly treated, which are then the carbamazepine, lanreotide, and venlafaxine. So those are then, there seems to be that the influent and effluent are very similar, and that might be one of the reasons why we're then seeing venlafaxine in the environment as well as we did with the previous study, that we were able to see that it seems to be bioaccumulating as well. So more work to be done in this part. Next slide, please. And here is mean, looking at just proportionally what kind of chemicals are coming through. And here we are seeing again the same as many studies overseas. Acesulfame K seems to be one of the top, having the highest concentrations in our samples. Venlafaxine is there. There's again carbamazepine. But then there's some newer ones like trickles, which could be then linked to the pandemic and the era that we are living in. But it's a bit too early to say what is driving these, and are these unusually about these concentrations really. But what I want to say that pay attention to the ends. I've done a number of samples on top of the panels. So some of them actually we were, and what I forgot to say, the x-axis is treatment train, from treatment train 1 to treatment train 9. So to some of the treatment trains, we have more replication, so they are not usually reading them entirely across. And if you look at the next slide, please, it's also about the different treatment trains. And then if you look at the mean concentrations, there seems to be differences, that there are some treatment trains that appear to be treating overall the pharmaceuticals better, but we need to look this into more detail to know then is that and what might be driving that. And next slide, please. But the good news is that there's more work that is underway, and we're lucky that the consultants that did the 2019 assessment will be now using our data to redo the risk assessment, and that will give us more information about the risks around these sites, and hopefully give better guidance for future as well. And then I'm excited that we are collaborating with University of Melbourne. So Dr. Mayumi Allison is doing for the bacteria test. And then Gerald Saruli and Professor Andrew Pasca doing toxicity assays, looking at receptor localization assay and ex vivo general culture assay, which will hopefully provide some more insights about the potential risks. And next slide. So in terms of then future, definitely long-term monitoring program would be ideal to get a really better understanding of background levels, because even though we're using passive samplers, there's still snippets in time that we're able to find evidence for. So longer term, visiting the same sites and getting an understanding of temporal differences would be best outcome. And for that, I definitely highly cannot underline enough that collaboration and creative partnership is the key. And we need to do more research to investigate the risks of pharmaceuticals to human health and environment. But what EPA is doing now and will be doing in the future, that we will be playing that role in developing better guidelines and environmental reference standards for pharmaceuticals. Thank you for your attention.